Hello, beloved learners. My name is Mr. Paul Chilola once again. Welcome to Black Slides Online. This is a platform where you have the privilege of learning the sciences for free. Okay, all you have to do is subscribe to the channel, and each time a new video lesson is uploaded, you'll be notified. Also, inform your friends that it is possible to visit this YouTube channel and learn entirely for free. The only expense perhaps is the time that you invest into watching these videos, and also maybe the internet data which you have to buy so that you are able to access the internet. I've always said that in case you have questions, don't hesitate to contact us on the numbers that are shared here. So welcome once again. We are going to look at a detailed description of food tests. This is one of the most popular experiments in Msce biology. All right, so I'll take you through a step-by-step procedure on a different testing for different food nutrients. Remember, food substances that we are going to be discussing are also called nutrients. Food nutrients. Food nutrients include carbohydrates, or in short called carbs, proteins, fats, stroke oils. Fats and oils together are called the lipids. Minerals, vitamins, as well as water. All these are called food nutrients. They are the components which we benefit from eating different foods. Each food nutrient has a role to play in our bodies. For example, carbohydrates, you'll appreciate that carbohydrates provide us with energy because they are rich in energy. Foods such as rice and sima, bread, potatoes, cassava, all these are called carbohydrates. We benefit from them. We get energy by consuming them. When you consume them, they provide us with energy, the energy we need to make movements, to walk about, to work, to do different body physical activities. All right. Proteins provide us with the essential amino acids, or amino acids in general, whose role is to repair our body worn out tissues, but proteins also provide us with growth. They help us to grow. Okay. Fats, oils, which are called lipids, together are essential for providing energy to the body's large amount of energy than carbohydrates, but they also provide us with warmth, apart from providing protective functions to some organs of the body. Minerals are essential for different body functions, providing us with the capabilities to contract and relax muscles, providing electrolytes to the muscles for contraction and relaxation. Vitamins are also essential for different body processes. Okay, water provides us with a solvent and a solvent environment in which different food substances, including air, oxygen, are dissolved. So water in the body plays a role of a solvent. So all these are called nutrients. So your syllabus requires that we should describe some simple experiments that we can carry out to detect the presence of carbohydrates such as starch. Starch is an example of a carbohydrate. Proteins, fats and lipids, fats and oils, which are collectively called lipids, some minerals, as well as vitamins, such as vitamin C, and as well as water. So in science, we want to clearly detect whether certain foods contain any of these nutrients. All right, so one of the food nutrients known as starch. Starch belongs to a nutrient called carbohydrate. Starch is a carbohydrate. Okay, we have other carbohydrates like glucose, sucrose, which is a common sugar, and others. Okay, so you find this starch quite often in foods like bread. Bread contains starch, cassava, maize flour, pasta, and the other foods. Okay, so you can carry out confirmatory tests to show that indeed these food substances contain starch. Also, those foods that do not contain starch, you can tell by carrying out same experiment. Depending on the results, you can deduce whether starch is present or is not present. So you as candidates, you need to be able to first to carry out the test and also to make inferences like deductions whether the food nutrient is present or is not present. So let's look at how you will test a food sample for starch. How can you tell that a given food sample contains starch? All right, so in order to test a food substance for starch, first and foremost, you add 2 milliliters of the food substance. Sorry, to 2 milliliters of the food substance, you add 3 drops of iodine solution. Iodine solution has a brown color, orange-brown color initially. Before you add anything to it, it has a brown or sometimes something closer to orange color. So to the food substance, whatever food substance may be, groundnut, cassava, flour, or whatever it is, you add 3 drops of iodine solution. Okay, then you shake and observe the color change. Remember that your observations are based on color change. Okay, so your observations on color change, there are 2 possible observations. If the color changes to blue-black color, if you see blue-black color, it means that starch is present. All right, so upon adding 3 drops of iodine solution to the food substance, to 2 milliliters of the food substance, If the color changes to blue-black, then it means starch is present. However, if the color remains brown, okay, in other words, if there's no color change, okay, it means that starch is absent. So if the food does not contain starch, the brown color of iodine will not change. Now, I want to warn you here that when you are recording your observations, make sure you record what you see. Don't say color changes. No. Record the actual color that you see. Don't say here, for example, don't say color does not change. No. You have to mention the color that you have seen. All right, so depending on the color change that you see, you can tell whether starch is present or is absent. If starch is present, then the color, the iodine color will change from brown to blue-black, blue-black. Make sure you describe this. Don't just say black or blue. You have to say blue-black. It means that the color is between blue and black. So mention the actual color that is seen. Okay? So if starch is present, the blue-black color will be seen. If starch is absent, brown color of iodine does not change, it remains the same. Okay? That's how you test a food substance for starch. In most cases, they don't tell you which substance is which. You are usually given labels. So you have food sample labeled X, or labeled Y, or labeled Z, for you to tell which one is or contains starch. So you carry out a number of tests on these samples. On X, you observe color change. On Y, food sample Y, you observe color change. Food sample Z, you also observe color change. So we are saying if the color changes, the brown color of iodine changes to blue-black, then it means that starch is present. And if the color remains blue, it means starch is absent. Okay, I hope that is okay. Let's proceed to another test, which is called reducing sugars. Usually, reducing sugars are what you call monosaccharides. These are monosaccharides. These are monosaccharides, or they're simple sugars. Examples include glucose. Glucose is an example of a monosaccharide. Okay, so to tell that a food sample contains a monosaccharide or a reducing sugar, you carry out this test. So to 2 ml of the food substance, you add an equal amount of Benedict's solution. Benedict's solution. Let's suppose that you have your food sample in a test tube like this, and maybe this is quarter away, quarter way, maybe a quarter of the test tube containing what? The food sample. That means you need to add an equal amount of Benedict's solution, which will make it half. So this is Benedict's solution. Very important. I mentioned in one of the videos that Benedict's solution is made up of 2 solutions, failing solution A and failing solution B. So when these 2 are mixed, they form a blue solution. Okay, so such a blue solution is called the Benedict's solution. So to 2 milliliters of the food sample or food substance, you add the Benedict solution, and then you heat the mixture to boil and observe the color change. All right. So there are 4 possible observations that you can make. If the color changes from blue to green, it means there is only a little amount of reducing sugars. Okay? So we are saying blue goes to green, indicates that there is sugar, there is a monosaccharide, there is a reducing sugar, but it's in a small quantity. If the color changes from blue to yellow, then it means there is just a little bit of reducing sugar, but more than when the color changes to green. So there is an average amount of reducing sugar. If the color changes to orange or brown, it would mean that there is a high amount of reducing sugar. Sometimes this color goes to brick red, like the color of a brick. If you know a brick color, it's red. Okay, that indicates that there is a high amount of reducing sugar. But if the color of Benedict solution stays the same, unchanged, it means that there is no sugar at all. There is no reducing sugar at all. Okay, so take note that there is no single result in testing for reducing sugar. You can have a green color, blue changing to green. It's an indication that there is sugar, but it's in a little quantity. Okay, if the color changes from blue to yellow, it's an indication that there is an average amount, just an average amount of the reducing sugar. If the color changes to orange, brown, red, brick red, then it shows that there's a high amount of the reducing sugar. In other words, there is high amount of glucose there, for example. But if the color stays the same and changed, then it means that the reducing sugar is absent. In other words, there's no reducing sugar in such a substance. All right, so I expect you to be able to explain this test or to carry it out in case they ask you, they present to you with different food substances for you to determine which one is reducing sugar. In the same way, they can give you samples like maybe P, Q, R labeled, P, Q, R, and S. So on each of these samples, food samples, you carry out this test to tell which one contains a sugar and which one does not contain a sugar, reducing sugar in this case. Once again, my name is Mr. Coach Lola. Please consider to subscribe to the channel so that every time a new video is uploaded, you are the first one to know. All right, another test that is also very likely that you can be examined on is what is called non-reducing sugars. In most cases, non-reducing sugars are sugars which are called disaccharides. Sugars that are called disaccharides are examples of non-reducing sugars. Example of disaccharides are sucrose, for example. Sucrose is the common sugar, like the loaf sugar, which we use in food, like making tea. It's an example of an non-reducing sugar. So we're saying sucrose. Sucrose is the common sugar, like the loaf one. Loaf sugar is a sucrose. Another one is maltose, or malt sugar sometimes. This is sugar that comes from germinating seeds, like maize seeds that are germinating. They have or contain a sugar called maltose. All right, so we can carry out a test to show that a given food sample contains non-reducing sugar. In other words, contains either sucrose or maltose. All right. So the procedure is not such a complicated one. To 2 ml of food substance, add 3 drops of hydrochloric acid. Okay. Okay. So let's suppose that you have sugar. You have a food sample that you are testing for sugar. So suppose this is food. So you adding to this, you adding 2 to 3 drops, or rather 3 drops of 3 drops of HCl. Okay. Then you boil. So having added the 2, you boil the mixture for a few minutes and let it cool. Few minutes, you can say, let's say 5 minutes. So if you have a stopwatch, you can record the time for which this mixture is boiled, let's say 5 minutes, okay, or less than that, okay. So having boiled the mixture for 5 minutes or below, you add the sodium hydrogen carbonate. So add the sodium hydrogen carbonate solution, dropwise until fizzing stops. Okay, so initially before you add the sodium hydrogen carbonate, you see fizzing, you see bubbles when you add the hydrochloric acid to the food sample. Okay, rather you see, some tiny bubbles. But when you add the sodium hydrogen carbonate, you see more bubbles forming, which signify that there is a reaction. Remember, hydrochloric acid, which is added, will react with sodium hydrogen carbonate. Of course, you can clearly see or tell that this is an acid. Sodium hydrogen carbonate is a base. That's in chemistry. So the products will be sodium chloride and water and carbon dioxide. So the production of the carbon dioxide will produce a fizzing environment, like bubbles forming. So we are saying add right. Sorry about that. So you add sodium hydrogen carbonate drop by drop until the fizzing stops. The fizzing is because of the carbon dioxide that is being released. So add an equal amount of Benedict's solution to the mixture and heat to boil. So you heat again. observe the color changes. So maybe they will ask you questions like, why do you have to add the hydrochloric acid? When you add hydrochloric acid to a disaccharide, the hydrochloric acid is going to break the bond, which is called the glycosidic bond, the bond between the molecules, the sugar molecules, all right? A disaccharide has two sugars that are joined together, like this. This is a disaccharide, a sugar, common sugar. So when you add the HCl, the hydrochloric acid is going to attack this bond. This bond is called the glycidic bond. Glycidic bond. So the bond will be broken. That's why you have to add the HCl. Okay. So this happens when the mixture is heated. Okay. But now the HCl added to the sugar is going to create an acidic condition. So to remove the acidic condition, you need to add a base. So the base is sodium hydrogen carbonate. So we are adding sodium hydrogen carbonate in order to neutralize the HCl, which is added in step number 1. So once the fizzing, the bubbling, or the effervescence has stopped, it means that all the acid has now been reacted by sodium hydrogen carbonate. Then you go ahead to add in your Benedict's solution. So remember that there are two heatings. There is a heating that is done in order to break the disaccharides into monosaccharides. And then there is also a heating that is done in order to bring about a reduction or an oxidation between Benedict's solution and the sugar that is available there. Okay. So once again, you also observe the color change. So there are two possibilities, or three, but for now we are going to limit ourselves to two observations, possible observations. If the color changes to green, yellow, orange, or brick red color, it indicates that reducing sugar is present. Remember that by doing all this, what we have done here, we are changing the sugar to the disaccharide to a reducing sugar. Okay, so a reducing sugar is made or formed when the HCl has acted on the glycidic bond. Okay, so it depends. If the color is green, yellow, or orange, it shows that there's a sugar present there. But if the color does not change, the color of the Benedict solution does not change or remains the same, it means that the non-reducing sugars are absent. It means the non-reducing sugars are absent. Very, very important. All right. Another test which you also have to be able to explain how to carry it out and the expected results is that of proteins, testing for proteins. Remember, proteins are made up of amino acids. The simplest amino acid is given by the formula NH2C, then you have an alkyl group over there and an H, and then a COOH. Basically, proteins are acidic. That's why they contain this, which is called the carboxylic acid. This is called an amino acid, the simplest one, right? So in order to test a food sample for presence of proteins, to 2 moles of the food substance, add an equal amount of 10% sodium hydroxide solution and shake. Okay, so sodium hydroxide solution is the NaOH. So you add this to the food sample, usually in a test tube. So you have a test tube. So you have a food sample. You have a food sample, and then to this you add the sodium hydroxide. You add sodium hydroxide. All right. So then you shake the mixture. Then you add in 1% copper sulfate. So you add 1% copper sulfate drop-wise and shake after every drop. Then you observe color change. Remember that sodium hydroxide and copper sulfate, they make what is called a burette solution. A burette solution. Burette solution. This is called a burette solution or burette test. Okay, it's a combination of 10% sodium hydroxide and 1% copper sulfate. Okay, so having done that, then you look for the following. If the color changes to purple, then it means proteins are present. Okay, another description is lilac. There's a lilac color. Lilac is a color that is close to purple. Okay, but just say purple color is seen, it means that proteins are present. But if the mixture still remains blue color of the copper sulfate remains unchanged, it means that proteins are absent. So take good note of that. Right, lastly, look at how you can test for vitamin C. Vitamin C, which is also called ascorbic acid. Ascorbic acid, vitamin C, can also be tested in the lab. So the procedure is a simple one. Do 2 ml of a solution or a chemical which is called the DCPIP solution. You add the food substances drop-wise. By the way, DCPIP is an abbreviation. It's a long name. In full, it's called dichlorophenoendophenol. Dichlorophenool sounds like a name of a drug. Endophenol. Let me write this in full. It's a long one. So we are saying DCPIP is short for dichlorophenoendophenol. Dichlorophenoendophenol. In short, is called the DCPIP. It's a chemical which is used to test for the presence of vitamin C. It is also used as an oxidation indicator. Oxidation indicator. Don't worry about this for now. You meet them as time goes. All right. So we are saying that to 2 ml of CBD, DCPIP solution, the full sample, you add 2 ml to the full sample drop by drop, then shake after every drop and observe color change. Naturally, the DCPIP is blue in color before you add any food substance to it. So if there is vitamin C, the DCPIP is going to be decolorized. Okay, so in other words, it is going to turn colorless. To be decolorized means becoming colorless. So this changes to colorless. To change to colorless. To decolorize is to remove color from something. Okay, so the DCPIP will change from blue color to colorless. Okay, that shows that there is vitamin C. But if there is no vitamin C, the DCPIP will remain bluish. That shows that vitamin C is absent in that particular food substance. My name is Mr. Pochilola once again. I wish you all the best in your practical exam if you have one coming ahead. And do your best. If you happen to face this question, I'm sure you're going to answer it very, very well. Consider informing your friends to join us on this channel and ask them, as many of them as possible, to subscribe. I'll be very, very grateful. Thank you very much for now. See you next time.